Alright, so welcome back. This is going to be our second screencast and probably our final screencast for Chapter 14. And just to recall, in Chapter 14 we are talking about the phylum Platyhelminthes. And remember, Platyhelminthes refers to the flatworms. And when we're talking about the flatworms, we're going to break this down into three different classes. And the first class is going to be the turbularians. And the turbularians are going to include the planarians. The second class is going to be the trematodes. And the trematodes will include the flukes. And the third class is going to include the cestodes. And the cestodes are going to be the tapeworms. Now, the first thing we're going to look at in this screencast is the nervous system that you would see in a member of the phylum Platyhelminthes. Now, these animals are going to be considered as having a subepidermal nerve plexus. Now, the word sub, of course, means below. And when you talk about a nervous system that is subepidermal, you have a nervous system that's going to be found right beneath the epidermal layer of these animals. Now, it's considered a plexus because it is a branching type of nervous system. Now, this nervous system is going to resemble the nerve net that we had seen in the cnidarians back in Chapter 13. Now, they have from 1 to 5 pairs of longitudinal or lateral nerve cords, and these nerve cords are going to lie right underneath the muscle layer of these animals. And so what you can see on the right here, and this is the planarian, is that these lateral nerve cords or longitudinal nerve cords are represented by this yellow line that you see right here. And you can see this yellow line extending over here. And if we're talking specifically about freshwater planarians, and again this nervous system could vary between the different flatworms, then we're talking about an animal that has what we call a bilobed ganglion brain. And bilobed refers to two. And so we have an animal here that has not one, but two lobes to its brain. And extending off of that bilobed brain, we're going to have a pair of ventral nerve cords. And again, you can see these nerve cords represented by this yellow line that runs down the length of the animal. Now, one thing I wanted to add real quick about this diagram is that in addition to the lateral or longitudinal nerve cords, we also have a transverse type of nerve cord as well. Now, don't get confused by the word transverse. Transverse simply means we have a connection that occurs between the lateral or longitudinal nerve cords. And so that's a basic idea as to how the nervous system is set up within this particular group of animals. Now, when it comes down to the sense organs with these animals, you're going to be finding that they have both tactile sense organs, and they also have chemoreceptive type of cells that act as sense organs. Now, if it's a tactile type of sense organ, we're talking about a sense of touch. Now, if it's a chemoreceptive type of sense organ, we're talking about being able to detect chemicals within the environment. Now, again, I'm going to continue to use the diagram that we had used previously of the planarian that you see on the right-hand side. Now, these animals have something called ocelli, and you can see the ocelli right here, and sometimes they're just simply called the eye spots of the planarian. And you can see right here they literally do look like eyes on this animal. Now, they aren't able to see actual images, but they are considered light sensitive, so they can pick up various levels of light within the environment. So in addition to the ocelli, these animals also have an abundant amount of tactile and chemoreceptive cells. And most of these cells are going to be concentrated in the auricles of the animal. Now, these animals sort of have a triangular shaped head, and the auricle is going to be found on each side of that animal's head. Now, in addition to the auricles, some of these animals will also have something called a statocyst, and these statocysts are going to basically function to maintain equilibrium in the animal. And what that simply means is that the animal is going to use these statocysts to make sure that it remains upright. Now, some flatworms will have rheoreceptors, and these rheoreceptors are going to be there to sense the direction of water currents. And so if you have a flatworm that is primarily aquatic, these receptors are going to be really important to that animal. Now, if you have a flatworm that is parasitic, they might have some special sensory nerve endings that are going to be found in the oral suckers or even the genital pores of those animals. And so it all depends on the type of flatworm that you're actually looking at, and that will determine what type of sense organs that you're going to find in that animal. So what we need to do next is we need to look at the reproduction and the regeneration capabilities found in each class found in the phylum Platyhelminthes. Now this is where things become a little bit more difficult because each class is going to have a unique way that they reproduce. Now the turbularians, which again if you recall are simply the planarians, their method of reproduction is relatively simple because these animals are not considered parasites. They are simply a free-living type of flatworm. Now when it comes down to asexual reproduction, they will reproduce asexually using either a fission type of reproduction or they can actually regenerate missing parts. Now when we talk about fission, we're simply talking about this animal simply constricting behind the pharynx and separating into two animals. Now when we look over here on the right hand side, you can see where this constriction is taking place, and as we had said, the constriction is going to happen right behind the pharynx, and you can just barely see the pharynx right here in this flatworm. Now remember, this is a planarian, so they have a mouth that is going to be ventral to this animal, and that ventral mouth is going to allow that pharynx to extend. Remember, the pharynx is part of that digestive system, so the constriction that occurs in fission in turbularians is going to occur right in this area. Now once the animal has separated, each half is going to regenerate the missing part. And so what that means basically is that you have this top portion right here that is going to be missing a tail. So it's going to regenerate that tail. And you have the bottom portion here that's going to be missing the head. And so it's going to regenerate the head region of that animal. Now this is going to provide for very rapid growth in populations. Now as we had said, these animals can also regenerate. If there's some sort of damage that has occurred to this animal, it can simply regenerate that missing part. Now on the right hand side, this is sort of an artificial example of regeneration in planarians, and in fact this is something you guys are going to do in class. We started off with one flatworm, with one planarian in this case, and a scientist had made very strategic cuts on this animal, and the result was a flatworm that has 1, 2, 3, 4 heads in one worm. So basically regeneration is pretty prevalent in the turbularians. So each end is going to be able to regrow any missing part. And again, this is something you guys will look at in lab. Now when it comes down to a sexual type of reproduction, and yes, these animals can reproduce sexually, these animals are considered monoecious. Now remember, monoecious is going to refer to an animal that has both male and female sex organs in one animal. Now what that's going to do is it's going to lead some of us to believe that they can actually fertilize themselves, but that's not the case. These animals still have to cross-fertilize, which means they still have to have a partner to reproduce. Now the turbularians will develop male and female organs that will open at a common pore. So there's not going to be separate openings for male and separate openings for the female parts of the animal. Now after copulation or mating, the eggs and the yolk cells are going to be enclosed in a very small cocoon, and this cocoon is going to be attached to the stalk of a plant, and when the embryos emerge, they're going to emerge simply as little adults. So there's not going to be much difference between the embryo or the larval form and the actual adult worm. So the next thing that we're going to do is we're going to look at the class Trematoda, and a little bit later on we're also going to look at the class Cestoda. And as I had said a bit earlier, when it comes down to reproduction with the parasites, we're going to be looking at a pretty complex life cycle that actually contains various larval stages. Now when you look at the class Trematoda, we're going to look at two different types of flukes. We're going to look at the human liver fluke, and we're going to look at the human blood fluke. Now the human liver fluke is one that actually is prevalent in the Eastern Asia part of the world, and so we're talking about areas such as China and Japan and areas such as that. Now the adults of the human liver fluke are going to live basically in the bile passageways of humans and even some fish-eating mammals. Now when you say bile, you're referring to the liver region of the animal. Now the eggs are going to contain a complete mericidium. Now mericidium is one form of larvae that will be found in the life cycle of this fluke. Now these eggs are going to be shed into the water with the feces from humans. And when the eggs hatch, they're only going to hatch when they are ingested by snails. Now, something that's unique about the flukes is that they actually have two different hosts. In fact, some of them can actually have more than two. The humans are considered the primary host, and sometimes we even call that host a definitive host, and the snails are going to be considered an intermediate host. And the eggs are only going to hatch when they are ingested by a specific type or genera of snail. Now the mericidium is going to enter the snail tissue, and then it's going to transfer into a second larval form called a sporocyst. And this sporocyst is going to produce basically one generation of a third larval form called a rete. Now this rete is going to pass into the snail liver, and it's again going to transform into another tadpole-like cercariae. So again, we have another larval form found within this life cycle. And these cercariae are going to escape into the water. Now once they escape into the water, they're going to burrow or bore into the fish muscles, or actually maybe even find their way under the scales of the fish. Now they're going to shed the tail, so they're going to transform again, and they're going to insist as metacercariae. Now a mammal who eats any type of raw fish, whether it's human, could be a dog, could be a pig, could be any other type of mammal, this cyst is going to dissolve, and the flukes will again migrate back up to the bile duct, and that bile duct is going to be associated again with the liver. And so basically the cycle will start over again. So the second type of fluke that we're going to look at is one called schistosoma, and this is called the human blood fluke, and you can see this fluke on the right hand side here. Now the eggs of this fluke will be discharged in human feces or urine, and once that feces or urine finds its way into the water, the eggs are going to hatch as ciliated mericidia. Now again, this is a larval form that's unique to the human blood fluke. Now they must contact a particular species of snail to survive. So this is another case where we have two hosts. Remember, in this case the human represents the primary or the definitive host, and the snail represents the intermediate host. Now once they're in the snail, they're going to transform to sporocysts, and these sporocysts are going to produce cercaria directly. Now in this case with the blood fluke, we do not have that rete stage. In the human liver fluke, we had the rete stage that was an intermediate between the sporocyst and the cercaria, but in this case we don't have that. So the cercaria are going to escape the snail, and they're going to swim until they contact bare human skin. And normally they're going to find their way into the human through the sole of the foot or maybe the ankle of the foot. Now the cercaria are going to pierce the skin, and then they're going to shed their tails. So these larvae are going to enter the blood vessels and migrate to the hepatic portal blood vessels. Now when we say hepatic, we're talking about the liver region of these organisms. Now they're going to develop in the liver, and they're going to migrate to target sites. Now these target sites are important because these are going to be the areas where the females can actually release eggs. And these eggs are going to be extruded through the gut or the bladder lining of the animal, and they're going to exit with either the feces if they come from the gut, or the urine if they come from the bladder of the animal. Now the eggs that remain behind become what we call the centers of inflammation. If you have eggs that remain in the gut, then you're talking about symptoms such as diarrhea. You could be looking at maybe abscesses found within the gut region. You could even be looking at ulcerations within the gut region. Now, if the eggs remain in the bladder lining of the animal, then you could be looking at obviously some bloody urine, and if whether or not it's in the gut or the bladder, both situations will result in some pretty extreme pain. Now, the very last class we're going to look at is the class Cestoda, and the only tapeworm we're going to focus on in this case is going to be the beef tapeworm. Now, when you talk about a beef tapeworm, you're talking about a tapeworm that lives as an adult in the alimentary canal of humans. Now, don't get confused by the word alimentary. We're simply talking about the intestinal region of the human. Now, the juvenile forms are going to be found in the intermuscular tissue of cattle. Now, mature adults can reach over 10 meters in length, and this 10 meters can be made up of over 2,000 proglottids, and we'll talk about proglottids in just a second. Now, this animal has a head region that is called a scolex, and the scolex typically has 4 suckers in regards to the beef tapeworm. But there's no hooks found in this tapeworm. If you're talking about a pork tapeworm, maybe a dog tapeworm, and there's various other types of tapeworms out there, some species do have hooks which allow them to become secure to that intestinal region. Now, when you talk about proglottids, oftentimes we refer to them as being a gravid proglottid, which means these proglottids, or these small sections of the tapeworm, basically contain shelled infective larvae, and these proglottids can actually be passed out in the feces of the animal. Now, each mature proglottid has muscles and a parenchyma layer, and it has male and female organs, so each proglottid essentially acts as an individual tapeworm. Now, these gravid proglottids usually will crawl out of the feces once they're released into the environment, and when they become dry, they're going to rupture. And when they rupture, these embryos that are released from these proglottids can become viable for up to 5 months, and they are typically picked up by any animals that are grazing within the environment. Now, to look at the life cycle of a beef tapeworm a little bit more carefully, it's important to understand that once the cattle swallows that larvae form of the tapeworm, they're going to hatch as something called oncospheres. Now, those oncospheres, that larval form of the tapeworm, are going to use hooks, and those hooks are going to be used to burrow through the intestinal wall, and they're going to find their way to either the blood vessels or the lymph system of the animal. And that's going to allow them to travel and reach what we call voluntary muscles. So these would be the muscles that we actually have control over. They will encyst in these muscles, and they actually become something called a cysticerci, or more commonly known as a bladder worm. Now, when the infected meat is eaten, like if we eat beef, for example, then the cyst wall will dissolve and the scolex will actually invaginate. And when it invaginates, it's basically meaning that it's going to attach to the intestinal mucosa within that animal. Now, these new proglottids that have been taken in by this animal will develop for a period of 2 to 3 weeks, and infected individuals will actually expel numerous proglottids on a daily basis after a period of time. Now, you can protect yourself from a tapeworm infection by making sure to thoroughly cook your beef. So if you're a person who likes your beef either very rare, simply bloody, rare, medium, you're probably putting yourself at risk for infection from tapeworms if you have an animal that has been infected. But if you make sure that your beef is cooked well done, or even very well done, then cooking at these temperatures can actually destroy those proglottids. So this will finish up our second and final screencast for Chapter 14. And as always, please make sure that you have completed the study guide that goes along with this screencast before you come to class.